Hello students, good morning to all of you. I am Dr. Srinivasan, Head Department of Microbiology. Today's session will be on antigen and antibody interaction, and I welcome you all for the session. Antigen and antibody interaction, as we all know, is an important process of immune elicitation in which an antigen is presented to WBC, and this WBC would function in eliciting an immune reaction resulting in the antigen antibody interaction. This antigen antibody interaction is as a result of various bonding that occurs between two molecules, that is the antigen and antibody, and this bonding can be anything from, as you can see from the slide, it can be from an hydrogen bond, electrostatic bond, or a van der Waals force or an hydrophobic bond. Each of these bonds are responsible for creating an interaction between this antigen and antibody molecule that further progresses into the development of what is called as an immune complex. Usually when an antigen enters into a host, there is a response called an infection cycle, and this is a result of an establishment of an infection. Due to that, the antigens establish itself, for example a bacteria, or it may be a virus, or it may be a fungal infection. It establishes itself inside the host and it elicits an immune response in our body. As soon as a WBC would interact with this antigen, that is the bacteria or virus infected cell or a fungal infected cell, whatever it is, this would mount an response, immune response, and these antigen antibody interactions are a sequel of immune response process that happens in our body. As you already know, there are several types of WBCs, and broadly these are classified into lymphocytes and granulocytes, and this antigen antibody interaction is made possible by the activity of what is called as your lymphocyte, that is the T lymphocyte and B lymphocyte. Next slide please. So the B cells, when they are activated in a response produced by the cytokines secreted by the T cells, they develop into what is called as the plasma cell and the B memory cell. These plasma cells are subjected to secrete different kinds of antibody which fall under 1 and 5 class, such as G, A, M, E and D, and any of these antibodies have what is called as an paratope in their protein molecule, and these paratopes are subject to interact with an epitope, such as the antigenic surface that are present on any of your antigenic molecule, that is your bacteria or virus or a fungal infected cell, or it can even be an inanimate object as well. So this interaction is made possible with another factor called what you call as an affinity. Please don't change. Affinity. So what is affinity? Affinity refers to the process by which the antibody and antigens are interacting at a particular point, or you call it as a domain, by which the strength of that affliction is increased or decreased based on the various factors that exist on the surrounding environment, and it is measured by the formula association constant called Ka is equal to K1 by K2, whereas the K represents the constant of affinity towards the antigen and antibody. And based on this, an antigen antibody interaction happens and the immune complex is processed. The stronger the affinity happens, the more the immune complex is present at a particular point, and this immune complex is subject to be activated and accepted by the immune cell and recognized by the immune cell. The stronger the immune complex, the stronger the affinity, the stronger is the immune complex, and when the immune complex is stronger, the immune response is also stronger. And another factor which determines the formation of this antigen antibody reaction interaction is your valence. We know the meaning for the word valence. What is valence? Valence is the empty space. In case of an atomic structure, we call it as the outermost orbital which has the electrons in it, whereas in this case, in case of an antibody, valence represents the number of empty spaces that are present, that are available for an antigen to get interacted. In other case, you can call it as the number of empty paratopes that are available for an epitope to get attached. This is present on the outermost part of your antibody, that is the FAB region of your antibody. Next. So now you can answer simple questions like what is a hydrogen bond, what is an electrostatic bond, what is a van der Waals force, and what is an hydrophobic bond based on the questions that are being prompted to you on your mobile phones. Like hydrogen bonds, anyone? Yeah, I get the answer from them. So hydrogen bond is the simplest bond that is being formed between two molecules, that is the two atoms, okay. And next is your electrostatic bond. Anyone for electrostatic bond? Yeah, exactly. Electrostatic bonds are the type of bonds that are formed between charged molecules. And next is the van der Waals force. Van der Waals force is a simple force of miniature force of attraction or repulsion between two molecules. And last one is the hydrophobic bond. Any bond that is dissolved by water is your hydrophobic bond. So any one or these, all of these bonding forces enable the interaction between an antigen and antibody molecule, provided the factors such as affinity and valency play an important role. Next. So based on the antigen antibody interaction, various types of immune reaction occurs in our body, starting from this precipitation, agglutination, third one is neutralization, opsonization, antibody dependent cell mediated cytotoxicity, and the last one is the complement activated membrane attack complex. So though these topics may seem as an individual happening within an immune response, all these are interconnected, and they finally, all these processes finally end up in the clearing of antigen at a particular point of time. Whether it is an acute infection or a chronic infection, an immune reaction starts with the activation of the WBCs and continues with the activation and secretion of antibodies and develops into the formation of immune complex, provided the factors such as valency and affinity puts its help into it, and at the end all these processes occur, and finally the antigen is eliminated from the body, that is the host. So these are the consequences of antibody binding. You can see here, being a microorganism, you have bacteria and viruses being neutralized by the antibody molecules. These antibody molecules, though they are represented as an individual molecule here, for example the Y shape, this doesn't mean that only one type of antibody is involved. You can have any class of antibody participating in an immune reaction. Soon after the participation of the antibodies and neutralization of the epitopes, you can have, you see you have different types of interactions, and you can have a process called agglutination or precipitation. What is precipitation and agglutination? The process in which you have a slightest difference in the reaction. In case of precipitation, the interaction between the antigen and antibody molecule is that antigen is soluble and antibody is insoluble in case of precipitation, and agglutination is the vice versa of precipitation, and this results in the formation of immune complexes. Both antigen and antibody interaction in case of precipitation and agglutination results in what is called as an immune complex formation. This immune complex, as you can see here, it's precipitated at a particular, or sedimented. Instead of saying precipitation, you can say it as sedimented at a particular area where the neutralization is occurring. Neutralization is happening when all the epitopes of an antigen is being satisfied by an antibody molecule. So when an immune complex is formed and when the neutralization process is completed, this immune complex is subject to be exposed to an attack by WBC. In this case, you can see a process here which shows as enhances, which means that an immune complex can elicit a phagocytosis process, which is the cell engulfment process. And when the immune complex is present, the phagocytosis is highly elicited, and more number of phagocytic molecules for phagocytic cells are invited to the site of infection, and the phagocytosis process occurs. And as a result, you can see the phagocytic cell are readily engulfing the immune complex consisting of antigen antibody complex. Next. So precipitation is the, as I already mentioned, it's a reaction between the soluble antigen and insoluble antibody. And this, when equal concentration of soluble antigens and antibody are present, there is a typo error, there is an insoluble antibody, okay, and equal concentration of soluble antigen and insoluble antibodies are present. you can have a structure like this. There is all the epitopes of the antigen and the paratope of the antibody will be satisfied, and this results in the formation of lattice. So as you can see, this lattice formation is the immune complex formation that occurs during any kind of precipitation or agglutination reaction, resulting in an immune reaction called the opsonization. If you have any queries or doubts, you can now raise your queries, or we can move on to this straightly to the question section, question part. So now we know what is precipitation, what is agglutination, and what is the antigen antibody reaction. You can raise your queries and clarify your doubts. Thank you for listening to the session.